Hi there, and welcome to a short video outlining antibody structure and function. So antibodies are protein-based, and it's immunoglobulin. So we often use this Ig as kind of a shorthand for antibodies. And the base structure is a Y-shaped structure. On there you have what's called variable regions, and those are out on the ends, and that kind of makes sense because that's where the binding is to the epitope. And then you have kind of the base structure, which are the constant regions. So because we know antibodies are specific to specific antigens or specific epitopes, it's that V region, that variable region, that's going to be altered, or not altered, but that's where all the differences will be, so that when the antigen comes in, it'll be specific for whatever that binding site is on that V region that's there. Okay, so antibodies are just a protein piece, so they're secreted by B cells, and specifically by B plasma cells. And again, the B cell system is often referred to as humoral immunity because it's secreted in the bloodstream or the body fluids, which used to be called the humerus. So often that type of immunity is called humoral immunity. And it acts in different ways, but in really the kind of big picture on all of these is it binds to the antigen and disables it in some way. So one of those is opsonization. So you'll actually see, and I'm not going to draw all of the binding here, but our antibody binding to the surface of, this is our antigen. I'm just kind of making a generic bacterial cell here. And it will coat it. And that attracts macrophages. And macrophages are going to ingest the antigen and then hopefully destroy it and then release it in fragments, so through the process of phagocytosis. So that's one function. There's also neutralization. And related to that is the antitoxin action as well. So again, same idea. So it's coating the antigen, but in this case, by coating it, it's not able to bind to the host cell surface receptors. So for example, if you were to have a virus, and it's counting on its spikes, right, to bind to a particular host cell for entry, but now it's coated with these antibodies, it can't bind anymore, so that's neutralization. And then antitoxin is also a type of neutralization. It just refers to not binding to the whole antigen, but the toxin that it makes itself. Okay, so that is a couple of activities. There's another one called complement fixation. Something separate. So we've got 1, 2, 3. And complement fixation actually attacks and lyses the antigen. So this would work well on a bacterial cell, for example. And then the last one is agglutination. And agglutination is, again, you have this binding that I drew over here, but it will cause the multiple or many antigens to clump together. So if you've got these bacterial cells and they're all coated in antibodies, they start sticking together and become this huge mass that's here. And that's going to, again, disable, whoops, that's going to disable the antigen, because if it's in this big giant clump, it can't bind to and attack your cells as well. So that's agglutination. Some of you are familiar with this, going back to the blood type example, which I've used in other videos, is you can actually elicit this response by mixing different blood types, and you'll see the clumping of the blood type that doesn't match. So if it's type B blood and it's type A blood that's put in there, you can get that clumping. Okay, so those are kind of the 4 actions of that. A couple quick things I wanted to mention are some of the subclasses. So IgG, that's the most abundant. And the Ig, again, stands for the immunoglobulin. There's IgA. This is found or secreted in the mucous membranes. IgM, which is an early responder. So often this is drawn in when you look at, you'll see it sometimes in inflammation drawings, because you'll see, you know, the steps of, you know, the chemotaxis and the stimulation of the immune system. And this is one of the early responders. And this is the one that has that kind of fun, I don't know if you want to call it a snowflake type surface there. So that's one of the first antibody to respond, at least in that initial infection with inflammation. Okay, and then there is IgD. And IgD is found on B cells. And then last one of my favorites, because everybody can kind of relate to it, is IgE. Maybe you're a lucky person that can't. But that's the antibody that's active in allergic response. And also parasitic worm infections as well. So again, you know, we've got these different types here. The general function, again, is then to bind to the antigen and disable it. So remember, the antibodies are protein-based. They do have some different subcategories, but to me, you could still sort of kind of group them together, because the antibodies are binding to the antigen and in some way preventing it, either by changing the surface through the clumping or the neutralization from getting the host cell, or maybe lysing the antigen directly, or also attacking more macrophages to the area, so enhancing that phagocytosis process. And then there's different subtypes that are there. You hear about IgG a lot because that is the most abundant. And then IgE, a lot of times you hear about if you're reading up on anything regarding to allergies as well. Okay, so that's just kind of a brief overview. It's a pretty complex system to kind of study and get into, but just gives you some idea of the basics of how they work and some of the different types as well. I thank you very much. Please contact me with any questions you may have. This is Corey Andres at Central New Mexico Community College using CNM Glass.